to the this thing. Coordinator, madam, you can go to the next this thing slide. We'll start now. Okay. Now this is the procedure how usually a simulation toolbox is prepared. Okay, that also implementing by this thing what you call machine learning or data science this thing algorithms. Okay, so let us see how it starts. If we increase the size of it. Okay, so this is an example of IL-13 pred. So this is a tool which will predict if any peptide can elicit interleukin-13 production from immunological cells or not. Okay. Understood. So in order to make this software, which is entirely based on prediction algorithms or machine learning algorithms. Now, how did they achieve? Is there any doubt? Somebody tried to ask something. Okay, so this is the example how Jain et al. Had produced this IL-13 prediction tool. Okay, now what this tool does, it will give you an idea whether any particular peptide can elicit production of interleukin-13 from immunological cells. Okay, mostly helper T cells. Okay, so I give you a little this thing that helper T cells are nothing but immunological cells which perform multiple activities. Okay, one such activity is to produce interleukins. These are nothing but cytokines which initiate multiple levels of immune response. It could be antiviral, it could be antibacterial, it could also be targeting cancer cells for preferential what you call degradation from the system. Understood? So if you Google no interleukin 13, what you will see is you will see that it's a pleiotropic interleukin. Pleiotropic interleukin means what you know. Pleiotropy is a genetic term. So in that, pleiotropy means a single kind of entity can produce multiple different this thing. What do you call this thing? Phenotypic effect. Are you getting? So pleiotropy means what? Pleiotropy means any genetic event or any entity which can produce multiple levels of effect. So if you Google simply interleukin 13, in simply it will say that it's a pleiotropic interleukin. Okay, it is not very specific, but it is pleiotropic. It can perform multiple level of activity. So we'll also see today this particular tool we shall study today. Now, how did they made it? Okay, so see here first and foremost, training data set. Okay, so we may not be computer science people, but we will have some idea so that we can direct our collaborators who will be from hardcore computer science how to make this thing suitable toolboxes for us. Okay, consider that you are a very good lab where your wet lab is excellent. Now what you are trying to do is you are trying to add one more layer to your this biomedical journey. So in that case, what you will do, the same thing that we are doing, we are trying to do. So collaborate with a computer science lab. Now in the computer science lab, you should give them a training set.
I think you are muted.
Hello.
Yeah. So
Deepnath sir, you are on mute. You are
Yeah,
not
yeah.
audible.
Okay. So you can ask me why we should know how to make the training set, because you are the one responsible who will be responsible for the overall efficiency of your tool. You may not be a computer science people, you need not to be, but when you are providing the training set, you will be you should be very cautious. Now see